Hi, good morning. Today through this presentation, I wanted to let you know about the regulation of the enzyme action. By the end of my presentation, you learn various methodologies that enzymes employ to control the metabolism. You can understand the various mechanisms present for the regulation of the enzyme activity. You also understand the mechanism of various processes like allosteric control and negative feedback inhibition, induction, repression, etc. So, why this regulation of the enzymes is essential in living organisms? We told that enzymes are very important for living organisms to finish off biochemical reactions. But there are several processes like photosynthesis, respiration, lipid metabolism, and other fatty acid metabolism, fatty acid synthesis, protein synthesis. Several biological processes are present in living organisms. Are all these processes are coordinated, or they are existing single like that? No, we can say that all are in coordinating with each other. How they are coordinating with each other in living organisms? By regulation of the enzymatic activity only, the various metabolic processes are coordinated, and they are also helpful in homeostasis, that means maintenance of the internal environment. That is why also the enzymatic regulation activity is needed for all living organisms. To maintain the internal environment, this process is required. So, there are generally two methods are there, that is for the regulation of the mechanism. That is, one is control of the enzyme quantity and the other one is altering the catalytic efficiency of the enzymes. So, these are the other things which will be affecting the control of the enzyme quantity and altering the catalytic efficiency of the enzyme. So, in upcoming slides we will go one by one. First, coming to the control of the enzyme quantity, it will be affected by the altering the synthesis of the enzymes. Enzymes are proteinaceous in nature, that means they are synthesized from the amino acids. So, with increase in the synthesis of the enzyme or degradation of the enzyme, the quantity of the enzyme is depending. So, the quantity of the enzyme is depend upon the either increased synthesis of the enzyme or degradation of the enzyme. Increased in the synthesis of the enzyme actually occurs when there is an increase in the reaction rate, that means when there is a need. Otherwise, when we do not use also, there is an increase in the rate of the enzyme quantity. For example, after taking protein rich food, there is arginase increase in the liver. That means the arginase is synthesizing very rapidly in the liver after taking rich proteinaceous food. But in the starvation animals also, there is an increase in the enzyme quantity because of the non use of the enzymes. So, there means there is no degradation. Here is an increase in the synthesis. In the starvation animals, there is no degradation. That is why altering the synthesis of enzymes or degradation of the enzymes, we can control the quantity of the enzymes. One more methodology, that is induction process. Induction is the process in which there is an increase in the enzyme quantity. That means these are the substances which increases the enzyme quantity are called as the inducers. So, based upon the response to the inducers, there are two sides of the enzymes called as the constitutive enzymes and inducible enzymes. Constitutive enzymes depends upon the concentration of the enzymes. They do not depend on the inducers, whereas inducible enzymes, they depend on the presence of the that means inducers. For example, induction of the lactase on the bacterium growing on the glucose medium is called as a inducible enzyme. So, by the induction also we can increase the quantity of the enzyme. The other method is the repression. It is opposite to the process induction. In induction process, we are increasing the quantity of the enzyme, whereas in repression, it is decreasing the rate of the synthesis of enzyme by a substances called repressor. Repressors are the molecules are having low molecular weight. Generally, they are produced at the end of the biochemical reactions. That's why it is also called as the feedback regulations. For example, take the dietary cholesterol, which decreases the synthesis of the HMG-CoA. HMG is a beta hydroxy beta methyl glucosyl-CoA reductase, which is the key enzyme in the cholesterol synthesis. So if you take additional cholesterol in the food, it affects the main key enzyme in the cholesterol synthesis. So this is the repression process, and the other factor which is affecting the control of the quantity of the enzyme is substances like substrates, coenzymes and cofactors also changes the conformation of the enzymes by decreasing the rates of the degradation. So there is an increase in the quantity of the enzymes. Likewise, depending upon the synthesis of the enzymes and degradation of the enzymes, induction, repression, and based on the concentration of the substrate, coenzymes and cofactors, we can regulate the enzymatic activity. And the second one, that is the catalytic, altering the catalytic efficiency of the enzymes. How this catalytic efficiency of the enzymes are occurring in the living organisms? That means first one is allosteric control. Allosteric enzymes are made up of more than one protein. Generally, they are having two sites, that is a catalytic site where the enzyme binds, and the other one is a allosteric site, means another site. So when there is a molecule, that means the molecules, if they bind to the allosteric site, if there is a increase in the synthesis of the enzyme, if there is a increase in the rate of the activity of the enzyme, then it is called as a positive effector or the allosteric induction. If effector is added to the allosteric site, then it is decreases the activity of the enzyme. Then it is called as a negative effector or allosteric inhibition. By allosteric regulation and allosteric inhibition, we can regulate the catalytic efficiency of the enzyme. Here is a figure for you where you can go through the positive allosteric regulation and negative allosteric regulation. So this is maybe due to the positive regulator brings the changes in the shape of the active site. When this positive regulator binds to the allosteric site, it also causes changes in the active site of the enzyme. So in that way it increases the reaction rate, whereas in negative regulator it prevents the binding of the substrate and decreases the reaction rate. The other mechanism is feedback inhibition. Feedback inhibition is an enzyme which that is the end product which inhibits the enzyme early in the metabolic pathway. In the figure you are looking at it is a simple feedback loop where the final product is interfering with the enzymatic activity in the early pathway. So this feedback control can be used for the decreasing the activity of the chromosome. There are two processes in the similar way, like negative feedback, negative effect and positive effect. Feedback regulation is there, feedback control is also there. Coming to the covalent modification, it is the modifications of the enzymes where by the formation of the covalent bond, the activity of the enzymes are changing. Covalent modification is reversible. That means a polypeptide chain is attached to the group by the covalent bond. So the other ones are zymogens or proenzymes. So zymogens and proenzymes are secreted into these tissues or cells in the form of an inactive form and can be activated later. So generally these zymogens are having extra polypeptide chain. If this polypeptide chain will be deleted from the zymogen, then these zymogens will become active. So these zymogens protects the tissues from autodigestion. There are several zymogens existing in the body like chymotrypsin, prothambrin, trypsinogen, pepsinogen, procarboxypeptidase, protein elastase and fibrinogen. Generally these zymogens become inactive either with the presence of the hydrochloric acid or with the enzymes or auto activity. That means by themselves they become active. Next is a covalent modification. So, covalent modification already we discussed, that means by the adding of the phosphate group to the substances, we can changes the properties of the enzymes or catalytic efficiency of the enzymes. And phosphorylation, this is the general process where it can be added, that is phosphate group can be added to the moiety of the protein moiety of the enzyme. By the addition of this phosphate group, it can be activated. It can also be deactivated by the removal of the phosphate group. This is called as the dephosphorylation, and addition of the phosphate group is called as the phosphorylation. Sometimes the phosphorylation is essential, and some for enzymes dephosphorylation. So, addition or deletion of the phosphate groups are also makes the enzymes active or inactive. And the final one, that is the protein-protein interactions. So, proteins are made up of catalytic sites and regulatory sites. So, here is a protein kinase enzyme made up of the two catalytic sites and two regulatory sites. So, in the form of a 2C and 2R sites, the pKa is inactive. By the addition of this cyclic adenosine monophosphate to the protein kinase amylase, so the 2R sites will be removed. So, by removing the 2Rs or two catalytic sites, the pKa will become active. So, these are the mechanisms. Through this mechanism, the enzyme activity can be regulated.